This image shows a microscopic field of rod-shaped bacteria. In a clinical setting, knowing exactly what type of bacteria is causing an infection is a matter of life or death, and doctors need a way to sort them quickly. That sorting is done using a chemical test called the Gram stain. It forces bacteria to take on distinct, unnatural colors, allowing us to categorize them simply by looking through a lens. This diagram illustrates the physical basis for that color division, the cell wall. Gram-positive bacteria, seen here, build their cell walls with a highly robust, thick layer of peptidoglycan. Gram-negative bacteria have a completely different blueprint. Their peptidoglycan layer is very thin, and it sits buried underneath a thick, protective outer membrane made of lipids. Before exploiting these physical differences with dyes, we prepare our slide. We spread the bacteria into a smear, allow it to air dry completely, and quickly pass it over a flame to heat fix the cells so they don't wash away during the chemical baths. With the specimen secured, the staining can begin. The colors that emerge at the end of this protocol will be a direct physical reaction to the structural architecture of the bacteria's exterior. Starting with the primary stain, we flood the smear with crystal violet for 30 seconds, then rinse. Both thick and thin-walled bacteria absorb the molecules, appearing identically purple. To lock that color in, we apply Lugal's iodine for 30 seconds. The iodine acts as a trapping agent, seeping into the walls and chemically binding with the crystal violet to form massive complexes. These new structures are simply too large and rigid to easily slide back out through the structural mesh. Both bacterial types are still visually indistinguishable. But because of their different architectural blueprints, the dye complexes are trapped in vastly different ways, setting the stage for the next step. Now we reach the critical chemical crucible, the decolorization step. We drip an alcohol or acetone solvent across the slide for roughly 30 seconds, and no longer. When the alcohol solvent hits gram-negative bacteria, it rapidly dissolves their thick outer lipid membrane. With this barrier gone, the remaining thin peptidoglycan layer cannot hold the dye, and the large purple complexes wash away entirely. The gram-positive bacteria react differently. The alcohol dehydrates their thick peptidoglycan layer. This causes the mesh-like structure to shrink and compress, securely clamping down on the large dye complexes and trapping them inside. If we stopped the procedure right here and put the slide back under the microscope, what colors would we see? Gram-positive cells remain deep purple, but gram-negative cells, having lost their dye, are completely invisible against the background. To fix this, we flood the slide with a counterstain for 30 seconds. The empty gram-negative cells absorb this dye, turning pink. The accuracy of this entire diagnostic test comes down to that brief decolorization window. Leave the alcohol on too long, and even the thickest walls will eventually lose their purple dye. Wash it off too quickly, and the thin-walled cells won't clear properly. After a final rinse with water to remove the excess pink stain, we gently blot the slide dry. We then place it under the microscope, using an oil immersion lens to achieve maximum magnification. If we stained a mixed sample correctly, the visual division is stark. We can clearly distinguish the deep purple spherical bacteria from the bright pink rod-shaped ones sharing the same space. Those colors tell us exactly what we are dealing with physically. The purple cells have the thick peptidoglycan armor, while the pink cells rely on their thin walls and lipid membranes. This chart summarizes the final logic of the protocol. Gram-positive cells have thick peptidoglycan and stay purple. Gram-negative cells have high lipid content and turn pink. The rule to remember is simple. Purple is positive, pink is negative. By mastering the strict timing of these chemical washes, we can reliably decode the cellular architecture of the microscopic world.